it's Peter here with my FSHD. All right, we got a great podcast for you, part 3 of our gene therapy and FSHD series. Now, actually, I know when I think about it, there could be additional parts as other gene therapy trials come online. But, in our first 2, we covered stuff, the nuts and bolts and what's important for gene therapy and what's known in this wealth of information from other neuromuscular diseases. But I know you guys love just hearing me talk. Right, no one likes to hear me talk, I know that. So I brought my true expert in everything CRISPR, and actually the person I rely on for so much, Cris Ameda, our CRISPR goddess. Cris. Well, I said I particularly need her here for this particular podcast, because not just to break up the monotony of me talking, take the heat, but there's actually a reason other than just, I enjoy your presence so much.
Witty banter.
And witty banter, yes. But do you remember, because it will pertain to the podcast, do you remember our first conversation way back in like 2011?
When I screwed up my gel shift and you were my last resort for help?
Yeah.
You're going to tell everybody about that?
Well, there's a point to it.
I hope so.
Well, you can tell everybody. You screwed up. I didn't screw up.
I had my PhD thesis required a lot of gel shifts. And
What's
for those of you who don't know, it's a way of looking at protein DNA interactions. So DNA binding proteins, you can see them on a gel based on the way they shift in oligonucleotide, which is a small piece of DNA.
So how do they see it?
Well, use radioactivity. It's really a pain in the ass to do. You need a license to use radioactivity.
So you use radioactive
you have
DNA.
to work behind a shield because these are, P32 beta emitter. It's really not fun. But if you do it every day, it's your bread and butter. You get used to anything. And I considered myself pretty good at gel shifts because I did them every day, every bloody day.
It was a great 1980s, 90s technology.
Yes. As he has made fun of me for.
But so the key is, right? You have your DNA sequence that you want, a specific sequence of DNA, and checking, does a protein bind this sequence? So you use P32 radioactive phosphate.
Label the DNA.
You make it super radioactive so that the DNA is incredibly radioactive. Now DNA that's unbound goes through the gel really fast because it just shifts, electrophoresis down the gel. But if something's bound to that DNA, it's
slower.
bigger and it goes slower through the gel and stays up at the top. That's why it's called a gel shift.
Right.
And so this way you can actually even purify transcription factors. You can use this
exactly
to monitor activities of DNA binding. And so it was really,
my whole PhD thesis was you have to identify this gel shift activity. What is this
I did some PhD,
figure out the protein that binds to this sequence.
I've done a few of these in my day as well.
Well, I didn't know that. And I thought
So
I was
what was
the
your
expert on
problem?
gel
So what
shifts.
was your problem?
Well, my problem was I was used to having slightly different equipment, set of plates.
Oh, you're
where
blaming
you
the
purify
equipment.
your probe.
You're blaming the equipment.
You purify your. This is the reason. And anyway, I had this.
This makes it sound like I didn't really tell her
It was an
anything that wasn't
absolute mess.
obvious. But
Absolute mess. And so I didn't know. Nobody there knew anything about gel shifts, but somebody said, "Oh, that Peter Jones, he's an epigenetics expert. That new guy that came. Hey, why don't you go ask him?" I was like, he's not gonna be able to tell me anything I don't already know. No one can help me. I'm screwed.
So the reality is, I never talked to her before ever.
Yeah, but
She's
I did go
kinda
to him
new,
because I was desperate.
and she just comes toddling over with this film. She's like, "So, I'll tell you,
You're the gel shift expert in
I
these
hear
parts.
you know a
you
little bit
figure
about
this
gel
out."
shifts. Well, you can figure this out." And she throws the film in my face.
And then, without batting an eye, in 2 seconds, he said, "I bet you ran the probe on these plates and."
You purified your probe on the same plates you used the gel shift
this
on.
giant signal is. It's the rest of that probe that got stuck to the glass and never gets washed off. And I was like, he's right. By God, he's right.
So
I better go work for him.
well, I did tell you something else, right?
You said you better get it to work now. Now we're invested.
Now I'm invested in you.
Come back in a week and let's see your data again.
Now I'm invested in you.
Yeah.
Okay, so she tells the story better when she's not being recorded, I gotta tell you. Because she's trying to cover her butt. The reality is
Trying to tell him in a coherent way.
it never occurred to her that just because she only had one set of plates and the lab was too cheap to do it right.
That didn't occur to me.
But
true.
the point is, the reason I bring this up
Right, make me look like a fool.
No, actually, that's when I knew that you were special genius.
Oh, really?
Yes, absolutely, because your response, as soon as I took one look at it, and as soon as I said, "Oh, you purify your probe on the same plates that," and instantly you recognized that was
It made perfect
correct.
sense. Made
That's
perfect
what you
sense.
did.
That's what
It gave you that
happened.
thing. You didn't argue. You didn't do anything, and you were just like, and so many people, when you say, "Oh, you screwed up,"
Yeah.
there wasn't really that much of a screw up, but it's,
No, it's
and you
just
also
one of those
realize
things,
you should have
yeah,
recognized it.
yeah.
But you immediately, it's just like, now I can move on to the next step.
Yeah.
And the bright light went on in your head. You understood that you had missed it. You understood you should have understood it.
Right.
You understood now you can fix it.
Yeah.
And you went off and got it to work. And what mattered was getting it to work and moving on to the next step.
Yeah, that's right.
Right? So different set of eyes on data, different set of eyes on a problem.
Always a good thing to have.
And your response was not, anger at me, or even a lot of times people you want to argue with you. Oh, that's not what happened. No. It's just it's about getting the answer, getting the right answer, and that's always what you've been about ever since I've known you. You put the egos aside, and you just get the right answer, and sometimes the right answer is in your head and it's all there. You just need somebody to just kind of pick it up.
Well, in science, you have to have to be willing to take criticism and accept it, but it's all for the good of the science.
But that was the other part about it. I knew it was all up there, and it was all right. Someone points it out and say, and we're gonna. So the reason I bring that up is we are going to be going over the publicly available Epicrispr Biotechnologies preclinical data that they have posted on their website that they presented at meetings and things, and there may be several instances. A whole lot of several instances where we might say, a different set of eyes on this data might say, maybe it's not working how you think it's working. Maybe it's not doing what you think it's doing. Or maybe you might want to reconsider some of it. Now, these are our opinions on the data, but I thought you were the perfect person but this is how our lab functions, right? We are harder on ourselves than anybody, make no mistake about it. But the reason we're doing this, and we did actually think a couple of times whether we should even do this or not, this podcast, even though people were asking for it, is, we do have a gene therapy company, Renogenics, and we're not wearing that hat today. We're not going to talk at all anything about Renogenics today, whatever. But one way to look at it is say, oh, these guys are competition. So should you really be commenting on your competitors' data? And it's I actually don't see any reason not to. Just a lot of people told us you shouldn't. It's kind of like the blue wall in police. It's like, should you shouldn't turn in a cop that breaks the law. Well, why not? Well, you just don't. Why?
We never have any regard for our own welfare.
The reason is they said it's going to come back to bite us in the ass. So that this will somehow again.
Sure it will.
But we are going to be very careful about review the data the same way that we would review a paper, right? Papers come to us all the time. You review a paper, you look at the data, see what they say. Does the data match what they say? Are the experiments done correctly on whatever your version of correct is? And, and you go back and forth. And that's the scientific process. Same thing with grants. Grants are a little bit different. You say, this is what I propose to do. And and this is kind of like a grant as well. We say, will this set of experiments answer the question that the grantee is asking, right? They say, I would like to answer this question. And they set up a bunch of experiments. And sometimes that set of experiments, in the best case scenario, is not going to answer your question. In which case, that's not a very good grant. Sometimes it will answer the question and no one cares. That's also not a very good grant. So grants are tough. But papers are, you get the data. And so, I've seen a lot of preclinical data from a lot of companies, and you can make these, they're all competing with us if we're thinking about us as a company. And oddly enough, we help a lot of them. Actually, when it comes to companies working on antisense, working on our mouse, we made the flex ducts for our mouse, we make the pig. We meet all the time with companies that technically are competing with, because we want them to get, because the point is, we want the right answer for the best technology to get to you, okay? That's the job. The job is not to get our technology to you. It's to get the best technology. If we have the best technology, hey, great, cool. But you know what? We want everybody to give their technology the best chance. And so one way to even think of this is that since we do not consult for Epicrispr, and we can't. We they actually asked, but so, that we do have an issue of direct competition with epigenetic gene therapy. We couldn't do it. But this is essentially the evaluation of the data that we would give. We're both consultants for Fulcrum. They chose not to listen to us as well. That worked out well, didn't it? And then the final reason that we're kind of doing this, and really kind of was what pushed me over the edge, is, I looking online and seeing the way, I have my own opinion of the data, my opinion of the trial, and we'll discuss that. And I will, tell you a little ahead of time, it's not very favorable. And that's my opinion and my interpretation of the data. But to see the way that this is kind of being fed to y'all online, right? Find myself saying, who's advocating for patients? Who's on your side? I see a lot of people saying, I've actually saw, from Mark Stone, for example, from the FSHD Society, that this is, life-changing hope or something like that. We'll talk about that at the end for families. It's just kind of like, I guess on some level, the hope that, you could conceptually say that hope, you got another clinical trial going, there's hope in that. But there's different ways to hope. You can jump out of an airplane without a parachute and hope that you survive. I hope it works, but you're better off with a parachute. Even better off not jumping out of the damn plane. But, there's just hope doesn't get you there, right? It has to be based in science. I haven't seen a single scientist come out and comment on the preclinical data or the trial, this gene therapy trial at all. Nobody's saying for it or against it, right? Everyone's just silence, radio silence, right? Now, granted, a lot of people don't like to put their name behind it, but when you see Avidity talking, right? They got Jeff Statlin, Nick Johnson, the top FSHD neurologists in the world are on board with them, talking about the data, talking about the trial. Their names attached to it. I don't see any name that I recognize attached to this. I haven't seen a KOL, that's a key opinion leader, say anything about that. I don't know if anybody knows anything about FSHD at all that's associated with this. When we look at the data, I just don't know. It doesn't appear to be the case. And based on my interpretation of the data, if it is, they need to get new people. Kind of tell you. So we're going to go through the data because you know what? Someone has to be on your side. Someone has to advocate for the patients, right? Someone has to be on your side. And that's good or bad. Because let me tell you, you guys have heard me on the Avidity podcast. I tell you, I like the Avidity trial. I like the data. I like the mechanism of action. In fact, I'll tell you what, I have seen a lot of preclinical data from Kate Therapeutics, from Armatus Bio, from Myricule. You know what? Great data. Really cool stuff. Excited about those trials coming. I've seen some others that are not so good. This potentially may be the worst. And that's just my evaluation of the preclinical package that's publicly available. Okay, so that's why we're here. We're going to just about the data. All right, we're going to go have talked enough. But first, go to Cure FSHD for All and sign up, because when we do get the next trial that's any good, what we're going to do is we're going to want to make sure that our non-ambulatory individuals, our teens and our preteens can get some access to the trial that's if it's showing some sort of benefit. And so they don't have to wait 5 years, 6 years till the end. And also to make sure that we get data on our most vulnerable to make sure that they're included in the label when we finally get FDA approval. So go to Cure FSHD for All and sign the petition if you haven't done it already. All right, we got some disclaimers. Myself, Takako, and Chris, as I mentioned, we're founders and we have equity in Renogenics, which is an FSHD-focused company with a CRISPR inhibition gene therapy program. We're also principal investigators of a research lab focused on FSHD at the University of Nevada, Reno. But that's got nothing to do with this podcast, because my FSHD and this podcast, we're not associated with the university. It's completely independent, not associated with a company, not associated with any individual, any foundation, or anybody. We're totally independent. And this podcast is for the sole purpose of educating the FSHD patient community about the science and the scientific process as it relates to the FSHD field. The opinions and interpretations and comments presented here are those of the speakers and are not endorsed by the University of Nevada, Reno, Renogenics, or any of our collaborators, any other entity, any individuals. It's just us speaking. It's our comments and our opinions. right? Remember that. These are our opinions and our interpretations on the data, okay? All the data is publicly available. We're not talking about anything that's confidential, so we may not have all the data, and that's fine. We're going to actually cover if there was more data, would that change our opinions? And I think you're going to find that the answer is no, but we sure hope there's more data. But the data is not meant to be all inclusive. It didn't cover everything, but covered what we consider to be important. It's not manipulated in any way. And this presentation does not represent medical advice and should not be used to make medical decisions. Just want to educate you on the trial, talk to your neurologist, and then decide whether or not you want to be a part of it or your loved ones want to be part of it. Either way, it's fine. We're just educating you. We're not giving you medical advice. All right, a little bit about CRISPR. All right, you are our CRISPR goddess. You want to tell us a little bit about CRISPR?
Well, CRISPR is as you say here, a natural system found in bacteria.
No, not everybody's watching online. We also have the audio. We got to make it a...
Bacteria evolved this really elegant system for self-defense mechanism, basically, so that when they are reintroduced to a virus, usually a virus, they can recognize it, say we've seen that before, they can cut it to pieces and get rid of it.
Get rid of it, right?
And microbiologists have been working on this for decades and very successfully. But some other scientists sort of saw the opportunity here that this was basically a tremendous potential to be a tremendous tool for gene editing and gene modification.
Well, basically because it's an incredibly specific, sequence-specific genome targeting.
Right.
Right now, the bacteria are targeting repeats that it has filed away to say these are dangerous repeats of viruses. Let's get rid of them. Yep. But you can harness that technology and target any part of the genome you want.
That's right.
So
absolutely, any part of any genome, not just human.
Any genome. That's exactly correct.
So really powerful.
But it was not the first genome editing technology, and I put this in here because that is always a theme in science. You always build. All science is derivative in some ways. And different people see different things and develop technology. And today's cutting edge technology
is built on yesterday's.
Which is now nobody uses. Right. So I'll just briefly mention this. So not the first technology, but it is the easiest to use. So originally there were zinc finger proteins discovered by Aaron Klug in '85. Carl Pabo actually, and the reason I know this pretty well, in '94, realized that you could link these are little DNA binding units that recognize a 3-base sequence. And you had a different unit for every 3 bases, and you take all the different 3 bases possibilities in the genome that there can be. Remember, there's 4 bases, you got 3 combinations, and you could link them all together and make a protein that will specifically bind a DNA sequence. All right, and then that was fused to this enzyme called FOC1, and now all of a sudden FOC1 introduces double-strand breaks in the DNA, and you could cut the DNA at any point in the genome that you wanted once you link all these things together. And the reason I know this really well is Sangamo Biosciences, which is still in business and still uses this technology. My postdoc advisor, Alan Wolf, actually left. He quit the lab. I was the last postdoc, and went to Sangamo to start them. Because the whole company built on this technology of turning genes on and off, editing genes, cutting genes, using this technology from the 80s and 90s. Yeah. Right, genome, because you can instead of FOC1, you could put an activator, you could put a repressor, kind of like everything we talk about with CRISPR. You could do that with this technology, and they actually have drug clinical trials going for some blood diseases with this, right? Yeah. But in 2007, something a little bit fancier came out called TALENs, right? TALES, T-A-L-E, is a transcriptional activator like effector, right? And these were natural factors found in bacteria. They were pathogens of plants. They were bacteria. Again, bacteria they have a lot of
amazing.
A lot of really cool tools.
Single cells.
Right. Right. And these factors bind DNA in a sequence specific manner. And in 2008, it was figured out that there's a code, and you could similar type of thing that if you take the different tails and you glued them all together in a row, you could target. If you put 6 tails in a row, you could target almost any sequence, right? Actually, with 6 zinc fingers in a row gives you 18 bases. The talons, each RVD, which is a repeat variable die residue for 1 base. So actually, you need 18 of them to make an 18. The reason I say 18 is that makes you unique in the genome if you go 4 to the 18th. And so, and again, so now people have been trying all this time, let's target the genome to cut it, right? 2008, they figured out the code. 2010, Daniel Voitis' lab figured out, fused FOC1, an enzyme that cuts DNA to the tails, made them a TALN, TALE, and the N is for nuclease, and now you could cut the thing. And actually, this is actually a technology that we started on in our lab, where instead of FOC1, we actually were going to fuse a DNA methyltransferase
that was the plan.
to the talons.
It was difficult and complicated. I was in the middle of it when the CRISPR storm broke.
Right, so we were actually doing this
Yeah.
for FSHD, doing talon epigenetic editing for FSHD on the D4Z4, what we kind of started, because it was a cutting edge technology. And here's a company that's still doing it, Selectus, right? And what a pain in the butt,
It was,
right?
yeah.
Oh my God, really complex
Yeah,
and
a lot of trouble.
expensive, a lot of trouble. And that's when the CRISPR
Yeah.
CRISPR news broke, right?
We dropped everything.
And we dropped everything. We just dropped the project. But interesting thing I caught when I was looking this up, Feng Zhang was part of this team, and he also dropped it.
That's cool, yeah.
Feng Zhang is probably going to
Sure
win
did.
a Nobel Prize for CRISPR, the second Nobel Prize for CRISPR, along with George Church. We'll talk about him in a second. But he actually was a talent guy. And then when he saw CRISPR, he also dropped it and said, you know what, this is a pain in the ass. CRISPR is going to be way better.
Yeah, this is the way to go.
All right, so as you mentioned, CRISPR's been around for ages, right?
Yeah.
So it was originally discovered in '87 by Ishino. He's generally credited with the first person to describe the CRISPR repeats. We didn't call him CRISPR.
Right.
Right, it wasn't until 2002 this Moeka called him CRISPR, termed him CRISPR. And in 2005, they figured out that these are kind of an primitive immune system in bacteria. Right, again, it's all about recognition of sequences. And that's always the key. Your genome, really, you have 4 bases, CTAG, and they're not in a random order, but if you start adding them together, how many combinations can you have? And you want to be able to target a specific gene in the genome, either to fix it, destroy it, turn it on, or turn it off. You need some very specific way to do it.
Right.
Because if it's not very specific,
And havoc, chaos,
you turn on
genomic instability,
genomic
disaster.
instability, disaster. Specificity is key.
Yeah.
Now we found when we did the zinc fingers and the talons, right? You actually did do some work with
I
that.
did,
It just didn't work that
It
well.
wasn't working that well. Yeah.
Didn't work that well, especially not compared to CRISPR. We just thought that maybe we'd try it just to make sure. But 2012, everything changed, and pretty much that's about when we.
Yeah, well, we saw this when it came out, and wow.
Right, so 2012 is when Jennifer Doudna and Emmanuel Charpentier published that they had taken this bacterial immune system and made it so that you can use it in the lab pretty easily
Yeah.
to do sequence-directed.
They turned it into a genuine
Genome-
tool for specific genome editing.
So that was what the Nobel Prize for, right? They didn't discover CRISPR.
Right.
They weren't the first to use CRISPR. But they were the first to develop it into a tool that
Right.
could readily be used
By just about anybody.
for just about anybody,
Just
even
about
us.
anything. Yeah.
So they won the
Yeah.
2020 Nobel Prize for the development of a method of genome editing. Right. And then I mentioned that Feng Zhang here and George Church probably won the next Nobel Prize in CRISPR because they were the first ones to show that you could specifically edit human cells. That was actually lacking from their previous.
Yeah.
Everyone was in plants and then other
Single word.
systems. Single words. They should have had human in
Words
the in
with
the
big
patent.
implications, yeah.
Okay, so we didn't discover CRISPR. We're just using it. So these guys made it so that everybody could use it.
Yeah.
Right? So CRISPR genome targeting and editing, right? So as you mentioned, it's by far the most versatile and easy to use of the gene editing technologies, right?
Yep.
You have the machine's the same for everybody. So for talons and ZFPs, you got to build a new machine for every sequence.
Right. That's right. For CRISPR, it's the same Cas protein, and you just need to, or ortholog, you just need to change your guide RNA. And the guide RNA will bind to Cas, and it will guide it to whatever location the genome matches the sequence of that guide RNA.
Right, so you have this little guide RNA here that's about 18 bases, finds a specific site in the genome, and it brings Cas, that's going to cut, or you can make a dead Cas, and it'll bring any activity you want.
Yeah, you can fuse a dead Cas that's incapable of cutting with whatever effector domain to do transcriptional activation, repression, epigenetic modifications. The toolbox is huge now. People keep expanding
huge toolbox.
the number of things you can do.
You're just using CRISPR for genome targeting to say, I want to bring this activity to this gene part of the genome.
Yeah, this location.
In any organism, pretty much, if you can get it in.
Yep.
Right, so it's a great tool for basic research, biotech, right? Uses for crops, fuels, microbe, livestock, and disease. Right, so it's really just having done zinc fingers, talons, and CRISPR. I don't know how many people have done all three. This is the easiest. I mean, this is just I don't know what the next thing's gonna be. I always think of like Men in Black, where they're just like, yes, this is gonna replace digital. Right? It's just like I gotta buy the White Album fifth time.
Eventually, it will catch up to sci-fi.
Yeah, well, it's genome editing. It's this is probably what I understand. There's a book out there about CRISPR.
Yeah, it's stuff you shouldn't do at home.
Chris Jones.
You shouldn't do it at home.
Garage biologist. Telling you, she is actually in her real profession, and she is a sci-fi writer, award-winning sci-fi writer, I might add. And CRISPR Evolution is already out, but your second book's coming out soon, right?
Yeah, should be out next month.
Check it out. It'd be really good. We'll get back. We'll talk about that when it comes. So things are always moving. So 1 year later, right? We had that was 2012, 2013. Stanley Chi and Jonathan Weissman came up with the dead Cas. That's where the dead Cas came from. The idea of, hey, instead of cutting everything, maybe you just want to use genome targeting. Of note, Stanley Chi is the founder of Epicrispr Biotechnology. So he's there, he knows, he's a CRISPR guy, definitely knows CRISPR, no doubt about that. Right? So first there was CRISPR interference. CRISPR interference is where you just take a dead Cas that can't cut, and it binds a sequence, and it just sits there, and it physically blocks transcription. So you gotta go DNA to RNA to protein. This prevents the RNA from being made by a physical steric hindrance. That's CRISPR interference. Whereas CRISPR inhibition, which was done 6 months later, is where you bind it to a sequence and you recruit in an activity like a repressor, and you actively shut down. So instead of steric hindrance, you're actively shutting it down. And then you came along.
Yeah, then we started doing this for FSHD. We thought it was the perfect disease to do a CRISPR inhibition
and it is.
Platform for. You have overexpression of a toxic protein, a toxic gene. If we can shut down this toxic gene using CRISPR dead Cas fused to a transcriptional repressor, that would be ideal.
No, we did also try DNA methyltransferase too,
we did,
right?
yeah.
It just didn't work nearly as well as a transcription repressor in our hands, right?
I have never been able to show actual methylation changes in fully differentiated skeletal muscle
as far as
cells.
we're aware, nobody's been able to show that still.
And trust me, we have searched the literature because this is a very important point.
So the devil's in the details. I love to say that because it's always true. It's right. The target tissue in FSHD in you is non-replicating, fully differentiated skeletal muscle. So DNA replicates, and one cell becomes two cells. Your genome gets copied, and it goes on. Though that's a very easy point at which to methylate the DNA or repress the DNA. You can always kind of activate the DNA, but to try to set up repression, it tends to be coupled with replication. That's where the machinery all tends to get in. But in skeletal muscle, once your satellite cells differentiate and go down the muscle lineage and they fuse into your existing muscle fibers, right? They don't replicate anymore. And that is a very different cell. And this is going to be very important when we get to the data. Because you got to make sure you're testing a therapeutic in the target tissue of your patient. And you did that. You showed that by the best way in our hands of doing it was using a transcriptional repressor to actively shut off DUX4 in the D4Z4.
No, that actually worked very well. Started with activation, remember? We fused the VP64 transcriptional activator because we thought it would be a lot easier to, just for proof of principle, can we actually activate And
So-
holy crap, it went gangbang. I mean, it
So-
does.
so-
Whew,
so
you ever
actually
want a
I-
lot of DUX4? We can do it.
Secret weapon.
Yeah. Because it's a lot easier
I
to
forgot-
activate something.
But I forgot about that's right because
That's normally
so
repressed.
the other the thing that people don't appreciate often in FSHD. So if you're working in Duchenne muscular dystrophy, actually in Duchenne, if you're a boy, you have one copy of the dystrophin gene. You don't even have two. If you're a female, you have two copies.
Right.
You know, two copies. But you're basically a single copy gene. Most genes, meaning your haploid genome, you have one copy of a gene. In FSHD, the DUX4 gene is in every D4Z4. You may have thousands of copies
Yeah.
of
And
DUX4.
for FSHD1, the vast majority are repressed.
Well, that's the other thing. In FSHD1, you might have 3 repeats on one chromosome 4, and you might have 100 repeats on the other chromosome 4, and then both your chromosome 10s might each have 100 repeats.
Yeah, and that's what you're measuring. You can never
And the sequences
specifically
are
look
close
at
enough.
just the pathogenic allele because the sequences are so close.
Right, so we weren't sure. Actually,
Are we
that,
going to be able to see a drop in repression in this huge background of repressed allele? You know,
so that was
the question.
actually I forgot about
Yeah,
that. Was
yeah.
actually a big point of this work was that you could actually use CRISPR to target the repeat
Yes.
genome.
Right.
So
That was the first time that
that's right.
actually the repeat genome had been targeted. I think
What
that was fair to
what
say.
people forget, 50% of your genome, your 3 billion bases, 3 billion bases of DNA in your haploid genome, 50% of that is repetitive DNA. People call it junk DNA, but 50% of that is repetitive DNA.
Right.
And
we had
so
no idea of this
so
before.
how no one had ever tried to use CRISPR or zinc fingers or talons
Yeah.
to target repetitive DNA.
Yeah, parts of the genome.
And so that's right. The first thing that you did is
Yeah.
you fused an activator.
Right, because I thought
So
that'd be a lot
actually-
easier to see, and it was.
So
It was a-
here, actually, so you had an activator, and the
Yeah.
question was, it's easier to see if you turn something on
Right.
than turning off. So it's easier to, so if you have something that's very low level, like
Yep.
1, 2, if you had 0 to 100, and
Right.
it's DUX4 is like at 1 or 2
Right.
on a scale of 0 to 100.
You can see 0, or you can see 1 to 100, but can you see 1 to 0.5? That's
Yeah,
gonna be
can
kinda
you
tough.
actually, so we didn't even know if it could work. So first thing you did is you put an activator, and you saw it goes to 100.
Yeah, and we thought, great, this system actually has legs
system
for
works.
FSHD. Let's try a repressor now. Let's see if that works.
That's right.
Yeah, and we
I
published
forgot.
all that in that first
That's
study.
right, and that's right. It's, see, that's why I have you here. Right?
Remind you of
Remind
historical
me.
details.
Yeah, well, because but that was proof of concept. You forget how as you science builds and builds and builds,
Yeah,
but sometimes you forget
all the steps along the way.
the complicating things, right? Because the first people doing CRISPR, there were 6 labs that I'm aware of that were doing CRISPR and FSHD, and all 6 were cutting the D4Z4, and none of them worked. So this actually worked.
Yeah, yeah.
And so what did we learn in our lab, in our research lab? That we could target the repeat genome
Right. We can
very
actually
specifically.
see repression.
You could activate it or you could
DUX4,
repress it,
right.
depending on what you fused to it.
Yep.
And the repressors work better than DNA methyltransferases in our hands
Yeah.
in the context that we use them.
Yep.
In differentiated skeletal muscle cells
Yes.
from FSHD patients that are the actual target tissue.
Thanks to Helen Blau's paper from the 90s, which described a very useful way of infecting
That's right.
skeletal
I
muscle
thought it was
cells. which-
80s even.
No, it was the 90s, I
Was
think.
it
I went to
90s?
look back and look, but a long time ago.
So Helen Blau is just a giant in the field of muscle biology.
Yeah,
She was
yeah,
at
for a long
Stanford.
time.
I mean,
This is maybe a paper that she's long forgotten amongst all her
but
hundreds of
again,
papers, but
devil's in the details. One of the reasons
cells are
people
hard to
don't
infect.
study the correct target tissue in
because it's tough.
FSHD is it's really
really
difficult.
difficult to infect.
It's difficult to infect and
Muscle
get your
cells-
stuff in.
Primary muscle cells are notoriously hard. You can't just dump
So-
a transfection reagent on them and expect your
So again,
DNA to get in there.
we didn't discover CRISPR. We didn't discover DCAS CRISPR inhibition.
No, it's
but
a matter of putting these things together.
We took it, used it, and then you didn't discover how to infect muscle cells.
Right.
You found a technology,
Right.
and you merged
You
the
merge all these
technologies
things to
together
get done what you want to get done.
to get it done and
Yeah.
get a result.
That is how most science actually is
if
done.
you're a good scientist.
If you're a good scientist. Now,
Crappy
if you're a true
scientists just
genius,
do-
you're the ones who win the Nobel prizes. You
True
make
genius
those
is
big
when
leaps.
that's actually
Yeah.
true,
Yeah.
because everyone sees the same thing and they see something different on the
Right.
same data set.
And
No,
thank
that's
God
absolutely.
for those people.
Thank God for
because
those people.
They move all of us forward.
But also you're a careful scientist. You're an incredibly careful scientist. And a lot of people, the advice Alan gave me, he's the guy who went to Sangamo to start Sangamo Biosciences. His advice to me starting out as a researcher, he said, find something difficult to work on.
Yeah.
You won't have much competition.
That is great advice, because everyone reaches for low-hanging fruit, and you
Easy
got
experiments.
tons of people reaching for the same apples on the lowest branches, and that's fine. You can do your career that way, but isn't it more fun? And isn't it more satisfying and
But
rewarding?
you get it right too.
To have the time to reach for the tough fruit that nobody's
The better
willing
fruit, to actually do something that's meaningful,
Yeah,
but
yeah.
also to do it right. Because
Yeah.
what I find is a lot of people do tackle hard problems and they do it wrong. They do it the easy way.
Yeah.
And that's how we became the tool makers for the FSHD space.
Some things are only worth doing right the first time.
We take the time to do it right? We've
and
often
this is
said,
one
and
of
we're going to
those
say
things.
this again, if you want to get there fast, go the long way.
Yeah,
Do the
yeah.
right, and that is how I train my students. If I were to ever have, I don't have students anymore, but when I train people, the mentality, what is the best experiment? Not what do we have in the lab,
Right.
not what do we know how to do.
What's
What
the
is-
question we want to ask, and what's the best way
What's
to
this?
address
Find a significant question,
Right.
and then what is the absolute best way to answer that, and then create the tools to answer it.
Yeah, I always appreciated that about you and Takako.
And
You do that.
And if it doesn't work, you can cut and you can,
Yeah, you could
well, because you did
cut
it the
bait,
best way.
but you did it
right?
the best
But
way,
you,
yeah.
you designed the right experiment with the right controls.
Otherwise, you can spend a lot of time banging your head
Well,
against
a
the
lot
wall
of
and
people
just-
try to force crap into a hole,
Yeah.
right? You can, it's not a square peg into a round hole. It's a square peg into a not quite the same size square peg.
you can spend a lot of
people
time
just
doing
spend,
that.
it's like, just do it right,
Yeah,
man.
just
do it right.
yeah.
Or sometimes you gotta change your question. Okay, so
Anyway,
recently you published this paper.
Yeah,
It's
yeah, some of our-
academic publication.
older data we finally
Finally published our
able to
old
publish
data.
it,
This is
yeah.
not renogenics data, by the way, just so it's very clear. We're never speaking. Where we got the whole CRISPR machine for the first people to. We'd actually done this years ago.
yeah, this is old.
But
stuff.
we to get it into the entire CRISPR machine into a single AAV. Now you'll remember if you listen to the gene therapy podcasts, right? The importance of getting everything into a single AAV, adeno-associated virus for delivery, because that's going to be very potentially dangerous, inefficient delivery. You're not going to have two viruses. You got to put one.
Yep.
And the way that Carice did this is she made an FSHD specific promoter that's very small and perfect for FSHD patients. We used a small Cas protein. She made a small transcriptional repression domain that,
Yeah, we use the small one.
and then was able to put the guide RNA in here.
Yep.
Okay, but just to be clear, this
not
is
our final
not
vector.
the renogenics
You're right, this is not
data
our
or
final,
vector
yeah.
for those of you who want to criticize us. Everyone has similar ideas. This is fantastic. You guys might know Mitsuru Sasaki Honda. He shows up at several FSHD meetings. He's in Hidetoshi Sakura's lab in Japan, and they actually published in bioRxiv, so a non-peer-reviewed journal in 2022, kind of what you were doing. And maybe got the answer on why. So it's funny. There's two ways to look at it. You could say they got it to work. They fused a DNA methyltransferase to Cas9, and they were able to get repression of DUX4, right, in FSHD. See how that, Now, the devil's in the details. One of the things about this is actually the cells they used were replicating cells,
That's right. Yeah. Yeah. These were iPSCs, and wouldn't
these
be
were-
surprised if you can alter methylation patterns in iPSCs.
So
with
these
stem
were
cells,
induced pluripotent stem cells,
yeah,
which
that
are
they
replicating like crazy.
then induced the myogenic lineage to become
So-
muscle.
so what they-
it's not the same thing as patient cells, primary, fully differentiated. Remember, your target tissue.
So what
fully
they
differentiated
showed
muscle.
is that if you took the muscle and make iPSCs, de-differentiate them into stem cells, then they could methylate, as these are replicating cells, methylate them with CRISPR, methylate the D4Z4s, then differentiate them in the lab back to make them into
Which is interesting,
skeletal muscle.
but not clinically relevant.
and they repress DUX4.
Right.
So that's not clinically relevant.
Right. But still interesting. This is they're one of the
Clinically
few
relevant
papers
is
on DNA
methylating
methylation
the
in FSHD.
differentiated cells. Nobody,
That's
to our
right.
knowledge, has ever shown
Yeah.
they can actually do
And
that.
we have tried because that would.
And
we
we
thought
have
that
tried.
That would be a good thing to do.
But cool work.
Yeah.
Definitely cool work. Love it. These guys are great. I'm not sure why the paper hasn't gone to peer review yet.
Yeah.
No idea. All right. So that's kind of our CRISPR branch. You can already tell this is going to be a long podcast, right? So
Yeah.
sorry, guys. I'll tell you ahead of time. You gotta jump halfway through it. Okay, quick refresher on FSHD relevant. Oh, you know what? Since we got a brief thing.
Yeah.
Speaking of, I'm always saying hi to people.
A shout out to somebody.
Yeah, Marietta.
Oh, Marietta.
Yeah,
Yeah.
we'll find out if she's living, right? Our friend Marietta,
Yeah.
Great FSHD researcher
Yeah,
who
FSHD research turned veterinarian.
became a veterinarian.
Yeah.
Absolutely.
Yeah.
She's
She still
smart
works for
move.
Davis, I think.
UC
But she
Davis?
is a listener of your podcast. She's
I've
a
heard that. You
regular.
never know who's listening. People from your past. We knew her at BBRI.
Yeah.
And, did some work that often gets cited.
Yeah.
On funky FSHD cellular phenotypes.
Yeah.
Yeah. Okay. The atropic, was it hers? The atropic
so,
phenotype. Yeah. She did that when she was over in France, and then came over to the U.S. and then just had enough with FSHD.
She was smart.
Oh my goodness! All right,
On to other challenges.
little other challenges. Marriage, for example, and kids.
Being a vet.
Being a
Juggling
vet.
everything.
I don't know
yeah.
which one's tougher. I'm going to go FSHD is the toughest. So FSHD, as you guys know, so those of you who do Zoom calls with me, and a lot of you have, know that I like to talk about FSHD. Describe it kind of like as a house. People want to conceptualize. The way I conceptualize FSHD, if a healthy muscle is like a house with all the bricks, a brick house, and all the bricks are good and solid, nice, strong house, can withstand a lot of stress. Whereas in DMD, every cell is missing dystrophin. And so every brick essentially is there, but it's not very strong, it's not built right, and enough stresses, it's going to collapse, right? And eventually the heart and breathing of the house collapses and the house dies.
Yeah.
FSHD is a bit different. In FSHD, all the bricks are there, and they're all basically built right, but every once in a while, one of the bricks expresses DUX4 and explodes, is a visual way to think of it, right? So one by one over time, if you're looking online at our YouTube, you see one by one, DUX4 expressing cell. And if you get enough of the bricks explode, the house can collapse, right? So again, you have a lot of healthy muscle to start with, and you start accumulating pathology over time, right? So it's a accumulation of pathology over time, right? Now, this is driven by the epigenetics of the region. We talked about FSHD as an epigenetic disease. And this kind of gets to the basis of the CRISPR inhibition, right? So you can have the same DNA sequence of the region as someone else in your family, but be very different in how you're affected. You could be more severe, less severe, asymptomatic, and you say, well, genetically, you are the same. But epigenetically, in respect to meaning DNA methylation and the histones and the chromatin, you're different, right? And so the less methylation, the less epigenetic repression, the more severe in general. And the more epigenetic repression you have, the less severe. And so you could actually think of it the way, this is what got us interested in FSHD therapeutics and mechanism. Nature has cured FSHD. Nature has said, if you epigenetically silence the region, as in people who are asymptomatic or very late onset, you don't have FSHD. The exact same sequence, the same repeat. Heck, even in an identical twin that's epigenetically different, you do have disease. So you don't need to fix the DNA, you need to fix the epigenetics. So the question is, can you reverse the epigenetics of the region?
Yep.
That's what Epicrispr Therapeutics is doing, or Epicrispr Biotechnology is doing, and that's what we're doing, right? And that's what the group in Japan is doing.
Yep.
And several other groups that are not publicly out there are doing, right?
Yeah.
This is the ideal disease for kind of epigenetic therapy, because the epigenetics dictate disease status.
Yep.
For the most part. There's always exceptions.
Always.
And you'll point them out to me and tell me I'm wrong.
No.
Not you, but somebody will.
Yeah, someone will.
Damn it. Yeah. It is funny. There are truly always exceptions.
Of course.
We've looked at epigenetics of over 3,500 people around the world.
Yeah.
Let me tell you, people are people. There's always exceptions, but in general, this is correct. And here's what it actually looks like. Okay, so if you're looking online, this is our epigenetic research test that we do as a potential diagnostic, depending on where you are in the world. And if you're looking online, you're going to see there's blue dots and red dots. A red dot is a methylated piece of DNA, a blue dot is unmethylated, and you're going to see about 25 places where you could be 24 places where you could be either blue or red, left to right. And you'll see that in a more severe case over here on the left, lots of blue, 0% methylation. You see at the less severe case over here on the right, you still have about 7 or 8% that have 0% methylation, but they have a lot more red, a lot more methylation. Right? So this is just an example of what exactly we're talking about here. The more severe, right, on average, at their Q2, they're at 16% methylation. So this is a contracted chromosome. Everybody is under 16%, very low methylation, severe FSHD. The less severe at the same level, they're at 45%. That's what is that, 3 times the level of methylation almost, right? So much less severe, right? More bricks are going to explode on this case than over here. It's the potential to express FSHD, and that's the wild card. The potential to express DUX4.
Yep.
And that's what I'm trying to show here on this next slide. If you're looking online, I really recommend looking at YouTube or looking at the video of the podcast, because it's not just that all the cells have equal potential to explode, expressing DUX4. All the bricks aren't the same. In fact, in a healthy individual, someone without FSHD, every once in a while, you do get a dysregulated cell that expresses DUX4.
Yep.
Right? We actually, Takiko published that in 2012. Everyone was kind of up.
Back when I first met you guys, this was one of your first.
That was one of our first. It was hugely.
It really blew people away because this was not part of the established model of how things worked.
The FSHD mafia tried to prevent this from getting published, actually, because the unifying model came out in 2010. Healthy cells, no DUX4. FSHD cells, DUX4.
Right, very simple, black and white.
Very simple, black and white.
Nothing's ever really that.
But every once in a while, we'd find some healthy cells that express a very small amount of DUX4, or very few cells in a healthy culture express DUX4.
Which makes sense once you understand it's an epigenetic disease.
And it was quantitative. There's so many more cells have the potential to express DUX4 in FSHD than in non-FSHD. But the fact that we blew the black and white binary idea, we actually had to really sometimes people question this, Jones, why do you guys take so long to publish stuff? Let me tell you, we got a lot of people trying to prevent us from publishing, and that is not paranoid. That's just the truth.
Unfortunately.
So anyway, so if you're looking online, these gray cells are the ones that are have no potential to express DUX4, and the green ones have the potential to express DUX4, the epigenetic potential. And individuals with infantile or early onset FSHD, most of the cells have the potential to express DUX4, and more cells are expressing DUX4.
Right.
It's kind of a stress response gene, and that's why the pathology starts so early and is more severe.
Right.
Other way to think of this is a weight hanging from a rope from the ceiling. You're cutting the rope, and eventually you cut the rope far enough that the weight drops. Well, in early onset, that cutting a lot faster because there's a lot more, the cutting, a lot more potential for bad things to happen. Now, in moderate methylation, you basically have it where, again, there's just fewer cells have the potential to express DUX4, and fewer cells are expressing DUX4. And in the high methylation, as you might expect, it's even fewer, right? And so so you are a mixture of cells, no matter everybody, you and I, everybody's a mixture of epigenetic signatures on a cell-by-cell basis throughout your muscle on potential to express DUX4 in response to either stress or stimuli, or we don't fully understand it, right?
Yeah, exactly.
Okay. Now, if you're going to come through with a therapeutic, and this is what's viewed as in this dark green, a therapeutic. Let's say you're going to come in now, if you have a really good therapeutic that hits all the cells,
then there's no problem.
No problem. Doesn't matter the potential.
Yep. But,
and if you have an antisense, it's probably going to go into all the cells probably pretty much equally.
Yeah, we would hope so. Yeah.
But an antisense technology like Avidity or microcycline siRNA, we'll call it, the question is, can you get a high enough level of your therapeutic in the cells so that when the DUX4 goes to get turned on, it gets shut off immediately? In gene therapy, it's different.
Yep.
You're trying to infect the cells with enough, so you need to infect enough cells with enough AAV to express enough of your cargo to shut off the DUX4 gene.
And remember, your muscles are big, and these are all the gene therapies are getting in through your blood vessels. So
which gets smaller and smaller as they get perfused, right?
Yeah, they're
So
not everywhere. So this is a tough.
so the question is, can you actually now get enough of the virus in and enough cargo to enough cells to go from a symptomatic to an asymptomatic case? You don't have to get them all.
That's right.
But where you start might matter.
Yeah.
Right? So where you start may matter. And so now you get a DUX4 positive cell pops up early on. Say if you only got 20% of the cells are infected, if you only are hitting about 20-25% of the cells because you have a very low dose gene therapy, well, it doesn't going to matter. You're still going to have popping cells popping.
Yep.
Right? It's going to be a little bit better if you go into a moderate methylation patient, and even better than for a higher methylation. But those people already are barely affected anyway.
Yeah.
Right? So you haven't really maybe changed
Yeah.
too much. Right? But the question is, let's say I just picked a number, 20%. Would a 20% increase in methylation matter? Because it's actually even less than what's going on here. You're still affected. You always think of this as a dial. Yeah, it's dialing it back, but it's still probably affected. But again, any level is potentially beneficial. So if you could truly change, but again, if you're at 0% methylation, they go to 20%, that's still FSHD methylation. You're really talking about these central ones here to try to get them into a healthy, right? So it's even more important to target all the cells.
Yeah.
If this is your approach where you're only going to increase methylation by a small amount, because it's only, people talk about going back to a healthy level of methylation. It all depends on where you start. Right? And then how many cells do you hit? Because if you're only going to take 10%, so if 20% of the cells increase methylation by 20%, you still have 80% of the cells potentially FSHD, right? Which, again, depends on where you're starting. All right, so how are you going to do this? Right, 2 options, right? We've just talked about.
DNA methyltransferase, which is the Epicrispr approach that we'll discuss when we get to their data, and transcriptional repressor, we've tried and other people have tried.
Yeah, so you can do it multiple ways. So the general idea, so this is getting back to what you kind of mentioned about, thinking, will it work? Right? Every one of these triangles, D4, Z4 repeat, has the same sequence for the most part. You can't target any sequence, right? But for the CRISPR amenable sequences, you have 4 repeat arrays that are the same.
Right.
Can you target the 2 or 3 or 4 repeats on the FSHD locus? And that's actually what you showed with the activation, right?
Definitely can.
So the idea is, here's your target sequence. If you're looking online, you'll see there's a target sequence in the repeat, but you're also going to see there's 100 target sequences. These are on targets. So you need enough of your repressor expressed at a high enough level to potentially, now we don't know if they're all accessible.
Yeah, there's the data in the literature is actually divided on that point. In some cases, some contexts, people have shown that, they have more trouble getting there. But all of these things are kind of dynamic, It's not like proteins bind and never let go. I mean, they kind of come on, they come off, there's all kinds of things going on in the cellular milieu. And so you always have to think of these things as dynamic interactions, and you want some interactions to predominate.
So you want a lot.
Anyway, the point is you want a lot. Yeah, exactly. Because you have to assume that all of these are accessible to some level.
Because the point is...
And I think a publication actually showed that it might take more time for them to be accessed in heterochromatin, but they do get bound.
Potentially it can be a sponge. Because again, if you're looking online, what you want to do is put your off switch, your CRISPR machine, on the contracted D4Z4 on the last repeat unit.
We really should have done that activation experiment on healthy cells to see if DUX4 could be turned on. That would have been an interesting...
You got time? What are you doing? You're goofing around with these podcasts.
FSHD non-permissive muscle cells, can you still activate?
Ooh, non-permissive.
Non-permissive. I want to see how accessible, and can you, maybe not non-permissive. Well, actually, never mind.
Yeah, we won't get into that. That would be actually, that actually.
We'll talk later. But anyway, getting way off track.
Exactly, because that one would be cool.
Yeah, okay.
For reasons we're not going to explain to you.
We'll probably edit that out. Anyway,
we'll probably edit that out. But the point is, if you're looking online, you potentially have all these binding sites, and so real low levels of expression might not be good enough for maintaining the off signal. So you want to have a lot of off here, right?
Yeah, exactly.
But you're right. That would be a good experiment. See, I told you she's a genius. She's brilliant. This is like our lab meeting, everybody.
This is our lab, since we don't actually have lab meeting.
This is why I don't have students. I don't need them. All right, now the truth is that the 2 different types of CRISPR inhibition are not going to necessarily work the same. There are conceptual differences, okay? So if you're going to target a transcriptional repressor like CRAB or MECP2, transcription repression domain, or any particular repressor where you're going to drop an off switch right here, where you're going to drop, the DCAS is going to bind, and you're going to actively shut off the locus, and you're going to maintain that there. So throughout, well, the life of the cell, right, and keep it actively off. Right, you're also going to have this off switch potentially everywhere else. Again, as Chris mentioned, it may or may not have accessibility, and these things kind of come off and come off. It's dynamic interactions, but conceptually, you're putting an active off switch. You're actively repressing. That makes sense. The DUX4 locus in FSHD, and so if for some reason this DCAS were to go away, you would potentially lose repression, right? This isn't necessarily epigenetic repression. Now with the DNA methyltransferase, it's a little bit different, potentially, right? The same idea, a DNA methyltransferase, because of the DCAS, of course they have the DCAS onyx, it's a different Cas, is going to bind to the D4Z4 and potentially all the D4Z4s and many other D4Z4s, which are already epigenetically repressed and methylated, right? Presumably it's going to epigenetically repress and methylate the locus at the FSHD1 contraction, right? Now, I put a question mark here because it's not entirely clear that just methylating the DNA or the chromatin at the CPG, just a couple of CPGs, is actually going to have any benefit. Now, if you epigenetically change the region and make it heterochromatic and actually turn it off, that's one thing. But it's not clear that actually just adding a methyl group is going to actually do the trick, okay? And the reason is that epigenetic states are set up and established during DNA replication in S phase. I'll talk about that in a sec, okay? So now the idea, though, just let's assume it's going to work. Assume you do epigenetically transform the region such that it's off. If the DCAS were to go away for some reason, the transgene silenced or something like that, that would stay off. So that's different than if you're targeting a transcription repressor, where if you have transgene silencing and you no longer express the cargo, you could re-express the locus. Here, if you truly did establish epigenetic repression at the locus, it should be maintained, even in the absence of the DCAS and the DNA methyltransferase, okay? But that's a big if, okay? And why is that? Well, all the talk from Epicrispr has been on DNA methylation, restore the DNA methylation. The underlying root cause of FSHD is loss of methylation. That is really superficial explanation of FSHD, okay? And I just want you, if you're looking online, look at these cartoons we got here. Chris has this great paper in 2015, a review that you did, another one in 2019. And what it shows you in a cartoon fashion, simplistic fashion, is an FSHD locus here on the right, a D4Z4. And a healthy D4Z4 here on the left. And you see that the difference between FSHD active DUX4 expressing D4Z4 and a healthy repressed not D4Z4 expressing locus is not just this little methyl group here or lack of the methyl group. There's a ton of other stuff, right? Epigenetic repression involves histone modifications. The histone tells us specific modifications, histone modifying proteins, heterochromatin protein 1, cohesins, SMCHD1, associated factors, all of these chromatin remodeling factors, as well as DNA methyltransferase, and the rDNA, small RNAs, polycomb repressor complexes. In fact, the whole point is about FSHD being an epigenetic disease, right? Is, epigenetics are different than standard gene regulation, right? It is actually a very stable, heritable chromatin state. This off state in a healthy situation is maintained. That's the point. It is actually very, very hard to turn this on. And say, well, it's on in FSHD. Well, that's because you were FSHD from the point when you were one cell or two cells if you're mosaic, okay? And so when this was established, both of these states are established during embryonic development. So if you have a normal size or healthy size D4Z4, repression is established during development in highly replicative cells throughout all the cells in your body, and then it is maintained, right? The DNA methylation is there, but all the histone modifications are there. All the repressive machinery is there. The chromatin modifiers keeping it off are there. All the accessory proteins are there. Everything is there. And then when the cell divides, it's all there again, and it's all there again. And that's what the methylation is doing. It's helping the cell remember that this methylation, this repressive machinery needs to reassemble after replication. Now, if you have FSHD and you're an FSHD locus, that repression doesn't get set up during development, okay? And so what ends up happening is it does not get marked for being off, and it can actually acquire euchromatic factors, different histone modifications, different chromatin remodeling factors, things like trithorax complex, ASH1L, the DBE10 long non-coding RNA. Again, you have ATPases, you have all sorts of things, CTCF, lambda, you have all sorts of factors that are associated with active or euchromatic or genes that could be turned on, right? They're not actively repressed. Genes that have the potential to be activated, and in muscle cells they are if it's FSHD. So it's a huge difference. Sure, there's no DNA methylation here, but essentially the model is, what they're trying to tell you is they're going to go from FSHD to non-FSHD by adding a couple of methyl groups, not even a ton of methyl groups, just a couple, just a small amount of methylation here and there on a couple of nucleosomes. And somehow all this activation machinery is going to disappear, and all this repression machinery is going to come in. And it's going to do this in the absence of replication. Okay? We know this happens during replication. So if it has any chance of happening now, again, epigenetics are set up to prevent this from happening. It's an epigenetically regulated locus. It is stable maintenance of off and stable maintenance of on. And it's really hard to flip the switch. Adding a couple methyl groups, flipping the switch, that kind of goes against everything that we know about epigenetics, right? So that's the question. Can this really be accomplished by adding a little methylation in non-replicating cells, or by recruiting a couple of repressor proteins? And now that's the caveat. DNA methyltransferases, like DNMT3A, the de novo methyltransferase, or DNMT3B, they are more than methyltransferases. They're also transcriptional repressors, and they associate with some of these other proteins. So you could imagine it's not the methylation that recruits some of these factors in. It's the actual methyltransferases themselves. And the methylation has methyl binding proteins. And so you can start to establish this, but again, it's during replication. Now, the thing to remember is, a lot of the work that gets done on methylation and these things, in experiments in the lab on naked DNA, on injecting DNA back in the late 90s, I was doing this. Back we'd inject DNA into Xenopus, the African frog called clawed frog oocytes, where the DNA gets chromatinized. It was a really great system, but you're taking naked DNA and assembling. If you methylate it, you assemble a heterochromatin. If it's unmethylated, you could assemble a euchromatin. Oh, so it just need methylation, but that's naked DNA. This is actually not, it's FSHD. This is not naked DNA. This is already epigenetically established as being on. Now, could you do that? I've talked about this for years, that is what we'd love to do, dial it back. But it's not just about adding methylation. It's basically, can you dial back the epigenetics? And the more I've studied this, the more I've been in it, I've come to the conclusion that's pretty darn hard, if not impossible, in vivo in a person. Doing it in the lab is one thing, in vivo in person, that's a different thing because you got to make sure with the cell. And that is why actually my lab has gone to targeted CRISPR inhibition with active repression. Okay. So what about these guys here? What about Sasaki Honda, right? This cool work. Well, there's some caveats to this, right? The caveats to this are, look over here in their graphic abstract. I never thought these would be useful. First off, they're replicating cells. Second off, they're not constrained by AAV, right? AAV says everything's got to fit within this 4,400 bases. So you need to screw with the Cas, make that small, regulatory region small, but also the repressor is small. My research lab uses a small transcriptional repressor, a small off switch, but Epicrispr is going to use a DNA methyltransferase. Those things are huge, very large, too large to fit into AAV. But Sasaki Honda was not constrained because this was just done in the lab. This was just done on a plate of cells. They actually have multiple Cas. They have dCas fused to a transcriptional repressor. So they're doing two things. They're showing that you have to have a transcriptional repressor, an off switch. So they're going to turn it off. And they're putting in two DNA methyltransferases, full-length protein, DNMT3A, and full-length DNMT3L, which is not active, but they have two of these together. So this is enormous. But this is more like the physiological state, right? So now you're putting this in, and you're actively shutting off the region. You have the methyltransferases, but they're also being, because it's full-length protein, they're able to recruit other repressive factors. So you're bringing in all of this machinery to the locus, and you're doing it during replication, where there's an opportunity as the replication fork goes through the DNA and displaces some of the histones and some of the other factors to give you an opportunity to reset what's going on at the locus by binding these factors there. And so when after replication, these factors are bound, they're now able, now all of a sudden, instead of being an active locus, you can say, hey, we're actually nucleating a repressive locus. Very, very different than what's going on if you're targeting a fragment of DNA methyltransferase 3A to a quiescent G0 locus, right? Okay, so quite possibly what's going to happen is that bring in a DNA methyltransferase, you might methylate a couple little spots of the DNA, might even methylate a lot of them, but it might still be epigenetically on and expressed. You might not actually have turned it off. So if you're assaying methylation, actually assaying methylation is not the right assay. Who cares, right? If it's because methylation of a euchromatic state doesn't necessarily, again, in the in vivo context of the genome, adding a little bit of methylation in a non-replicative state, is that going to flip the epigenetic state? Does anyone think it will? Is there any data that says it will? Not that I'm aware of. All right, so as you mentioned, it's got to get to muscle.
Ah, yeah, I love that picture of muscle.
So everybody starts out with cells in 2D growing in a dish and still cheats the system because they're doing replicated ones. The reality, the target tissue is non-replicative,
right,
differentiated, fused, three-dimensional.
Fused, vasculature is there, it's innervated, there's immune cells running around. I mean, this is the real
and you have to get your therapeutic through the blood to the myonuclei, which may or may not be centralized,
right,
and expressed.
Yeah.
Avoiding all the immune cells, avoiding all the garbage, the potential, the extracellular matrix, and other things that can bind it up.
Yeah.
It's got to get into the cytoplasm, which is all this red part. If you're looking online, it's got to get into these blue dots here. These blue dots are the myonuclei, which is a very small fraction of the muscle.
Right.
Right. So it just gets into the cytoplasm, and it's not going to be expressed.
That's right.
And so you could actually get a lot of virus in, but not all of it's going to necessarily.
Or it hangs around and gets stuck in the extracellular matrix. I mean, there's a reason why viral doses are always so high.
Right. So, which is why you want to make sure, right, you want to have a high dose that's safe.
Right. Which I think you talked about in your other.
We talked about in the other podcast. We're going to get back to it. So the way to do that is in vivo versus ex vivo. And again, just want to remind everybody, when you hear about CRISPR or gene therapy working in the news, a lot of times it's ex vivo, meaning a lot of times the disease is amenable to taking cells out of the body, fixing them in the lab, and then putting them back. This can be liver cells, this can be blood cells, this can be cancer-fighting CAR T cells,
Yep.
right?
It's a lot easier prospect.
It's a lot easier
without
prospect.
trivializing it.
Or
It's a
you
lot
might
easier
actually hear
than
about
in vivo.
fixing a percentage of the cells in the liver.
Yeah.
That was a recent one.
Yeah.
Mainly because everything you inject into a person ends up in the liver
Yep. Yeah.
at some point, some percentage of
It's
it.
a nice target tissue to
And
have.
it's very regenerative, and you only need to fix a small percentage of them to actually get benefit.
Right.
But for FSHD, we're going to need systemic AAV delivery,
Yeah, to body-wide skeletal
right,
muscles.
to all the skeletal muscle in your body. And again, a lot of it's going to want to go to the liver and to other organs, but you only need it in the skeletal muscle.
Yep.
Correct? All right, I'm doing that with AAV. Right? And this is where you earned your
Stripes.
I was gonna say, the first proof of concept, I forget, that first paper really was impressive.
What, the CRISPR paper?
Yeah, I mean, really, to show, because I had forgotten really how that nobody knew you could target repeats at the time.
Yeah.
We were the first person to target the repeat genome, right? That's actually hugely important.
Yeah, we had to look because some reporters were trying to give us credit for things we hadn't done, and we were horrified, remember?
Well,
And
but
so-
you had done that.
Yeah. We had to carefully look
Well, then
at the
that's right, they
literature
actually said
and
that-
say, what can we actually say that we did that was new?
You
or
got
unique?
credit for coining the term
Yeah.
repeat genome, but Jeannie
That's Jeannie
Lawrence,
Lawrence, yeah.
actually coined the term repeat genome.
Yeah.
Anyway.
Anyway.
But most recently, your big feat has been to fit everything into a single AAV. And actually, Epicrispr, their biotechnology, I always want to call them therapeutics. Epicrispr Biotechnology also fits everything into a single
Right,
AAV.
right.
Modalis must
Which
fit
is good.
everything into a single
Yeah,
AAV.
people realize this is important.
Right? And so how are you going to figure out if this works? Well, we have 2D and 3D muscles. You have the FlexDux4 mouse available. We have human xenograft mouse models. We now have an FSHD-like pig, and then you have non-human primates. So we have all these tools, and you can test if these capsids, if this virus can get to enough of the cells in the body to a single IV injection. And you can learn so much before you go
People.
to a patient.
Yeah.
In fact, I think that's our duty as scientists and members of the FSHD community, is to not just de-risk everything. When I think about de-risk, everyone thinks about toxicity and safety. To me, de-risk means we're not going to do an experiment in a patient that could have been done in a dish or an animal model, right? Because the patient is actually the person that they're at risk. And even if they don't die or they don't even have adversity, they're giving skeletal muscle, they're giving biopsies, they're giving their time, they're giving their mental effort.
Yeah, and maybe their ability to participate in other trials too.
And their potential to participate, that the idea of taking something into the clinic that has very little to no chance of benefiting the patient, to me, is unconscionable.
Yeah.
If that were to be done. And secondly, to go in with something with questions that could have been answered
And that could have been answered in another
system, which
yeah.
essentially, are very, essentially can almost kill the technology, right?
Yeah.
Right? So we would never do that.
No, we're doing all this
Damn right. Damn right.
one after the next.
All right, so there's, I tell you, devil's in the details, right? So remember, there's these are the three publicly disclosed companies: Epicrispr, Modalis, and Immunogenics. Different Cas9 proteins, different repressors, different guide RNAs, different promoters, and different capsids. The one we know the most about, of course, because they're in the clinical trial, is the Epicrispr Biotechnologies. They're taking AAVrh74 and DNA methyltransferase and going into people, right? All right, we're gonna take a break right here and grab yourself a coffee or something. I know Chris needs a break, but just sum it up really quick before we go on to the next one. Just again, FSHD is complicated. It's different than the other neuromuscular diseases. Dominant gain of function, small number of cells expressing the toxic gene. A lot of cells having the potential to express the toxic gene, but not all of them. And really the pathogenic gene expression is in fully differentiated skeletal muscle, not replicating cells. And this actually really does matter, okay? It really matters. And so I think that kind of maybe what's new from some of the previous podcasts, if you're an avid listener, is I just wanted to make sure everybody's clear that while we talk about methylation a lot, right? And in healthy or non-FSHD cells, heavily methylated, hypermethylated at the D4Z4 locus. And in FSHD cells, it's hypomethylated, unmethylated, low levels of methylation. And that's diagnostic, and it tells us a lot. But these are, just one of the modifications of the locus. Epigenetics is not just DNA methylation. There's all the histones that the DNA is wrapped around the chromatin, and these histones have active marks in FSHD and repressive marks in non-FSHD. There are small non-coding RNAs that maintain activity or repression. There are chromatin remodeling enzymes that keep it active or keep it repressed. There's additional repressor proteins that are recruited. There's additional activator proteins that are recruited. There's this whole, kind of conglomeration of euchromatic, active, potentially expressed factors that keep the locus open and gives it the potential to be expressed in FSHD. And conversely, there's just a whole number of chromatin factors that assemble at the locus in a structure that keeps it repressed. And the idea that just changing a couple of methylation marks at a few spots. Now remember, that's the other thing to keep in mind. Adding a little bit of methylation, we already know we have a lot of active FSHD chromosomes that have a little bit of methylation and they're still active. And you don't have to sound zero or little one methylation makes it inactive. So it's not clear how much you need to add to turn it off. But it's not even clear that adding any actually turns it off, because when these states are established as early in development, the epigenetics at the FSHD region, if you're not FSHD, established as repressive early in development, and if you have FSHD, this was also established early in development. Okay, you're just several thousand cells. These things get established and then maintained all the way out through development. And the whole point of epigenetic gene regulation is that these states are incredibly difficult to flip. Otherwise, it's not epigenetic. It would just be normal gene regulation. The point is they're heritable, stable states that, sure, under certain circumstances can be flipped. But in general, off stays off through multiple generations and on stays on through multiple generations, right? And so this sort of idea that you can just change a couple of methyl groups on the D4Z4 and suddenly all the proteins and all the chromatin modifiers and all the histone modifications that are associated with this large active region suddenly are gone, and now all of a sudden all these repressive factors are brought in. It's just highly unlikely, especially when the cell's not replicating. In fact, the only chance you'd have really of that happening is if the cells were replicating, because during replication, these factors actually have to kind of get removed from the chromatin. So the replication fork can go through, copy the DNA, right? The DNA polymerase comes through, copies the DNA, your maintenance methyltransferase, but all the factors are kind of there, but they kind of have to dissociate and come back. And so you kind of blow up the system and then reassemble it, right, during replication. And we know actually even in de novo methylation, right, when this actually happens during development, de novo regions, or in cancer, where you're suddenly getting methylation where you don't want it, de novo methylation, these are highly replicative cells. There's lots of DNA replication going many generations, and that's when you change the methylation status. This idea that in a cell that's not replicating and the genome's just sitting there and an active or euchromatic region is there, and suddenly it's just going to throw a couple of methyl groups on there and suddenly it's a repressed, off region. How does that happen? Is there any evidence that happened? So sure, you might even be able to detect, hey, I added a few methyl groups. Does it matter? I actually have a hard time. I've never seen any data that it does matter in a non-replicative. So these little details matter, because how you do the experiment's going to matter, because we know what's going on in you. Your differentiated skeletal muscle is not replicating. You're in FSHD, you're in a euchromatic open state. And if you don't have FSHD, it's repressed. And this concept that it's going to suddenly flip really easy and then stay that way is, well, I don't know. I haven't seen any evidence that that can actually happen. And that's really important, because when you want to think about the fundamentals of a therapeutic, right? And that's something I always think about all the time. That's why I'm trying to educate you all. And whether it's siRNAs, antisense oligos, heck, we'll talk about stem cells. We can talk about anything. We can talk about lesmapimod. Understand the pathways involved, understand the mode of action, understand the thought behind it, the science behind it, and then the evidence. Because you've come up with a hypothesis, and that's actually the case, right? So that's fine. The hypothesis for the Epicrispr trial is going to be that in a non-dividing, fully differentiated G0 cell, that adding a little bit of methylation, a couple of methyl groups here and there at some point in the D4Z4 is going to somehow lead to the FSHD region being off, and enough of this is going to happen that enough cells are going to be off that it's going to be therapeutic. Again, that's very different than looking at siblings where you have an asymptomatic and a symptomatic sibling, because their states, even though they might only be different by, 10% methylation or 20% methylation, the epigenetic states were established during development. So it's the whole epigenetic status and the whole population of cells that have this, and these are the averages of all the population. So it's going to be, how many cells do you get to? Also, it's going to be, does your modification actually matter? And you need to test it in the right system. So in the next part, we're going to go through and look at all the data and see, okay, they have a hypothesis. Let's see how they did testing it. Let's see what their data says. Okay, it's fine to have the hypothesis. Hey, we think we can change activity state by changing the methylation. The hit and run paper by the great group in Japan, Sasaki Honda and that gang, right? They showed you can do it in highly replicative iPSCs. That's not the right tissue for FSHD in vivo gene therapy. So we got the hypothesis. Hope you understand it. Join us in the next part and let's see if it works. I know where my money is.
Thank you for listening to the MyFSHD podcast, where we share with you the latest news and information on FSHD as we strive for a cure. You have a question for Peter? He'd love to hear from you. You can reach him at peterjones@med.unr.edu. Thank you for listening, and join us again next time for more news and information.